Thanks, Tim, for that wonderful introduction. And I'd like to thank the organizers for the invitation to come and present here today. So today I'm going to talk about HIV vaccines, where we are now, and I'll take a leaf out of Nyaradzo's book and talk about where we've come from to give us a way forward to where we're going. I have no disclosures I have nothing to disclose right now and no conflicting interests. So my presentation outline is to talk about HIV vaccines, why a vaccine is important, development of BNAbs, broadly neutralizing antibodies in HIV infection, how to induce these, and lastly, I'll talk about BNAbs and look into the future. So as Tim has just said, yes, we've had lots of trials, more than 100 phase 1 trials since 1987, and less than 10 efficacy trials in the HIV field. And RV144 that is shown here was the first trial to show a glimmer of hope, well, to show that there was some efficacy that it actually showed at 1 year after of follow-up. And we saw that this was 61% efficacy. And then at 3.5 years, 4 years, there was stabilization at 31% vaccine efficacy. Since we hadn't seen anything like this before, we're very excited, and the correlates of protection showed that non-neutralizing antibodies were the ones that were contributing or were responsible for this reduced correlate of risk. And then this spurred the next trials, which was HV10702. Actually, there were many smaller trials, phase 1 trials before this, but then HV10702 was the biggest trial where we looked at the same immunogens that were used in the RV144 trial, but now trying to make it more applicable to the subtype C group. So we actually changed the immunogens from an AE that was circulating in Thailand to subtype C circulating in South Africa. And this was meant to make the vaccine more immunogenic and more clade matched. Again, what we saw was that there was no benefit in terms of HIV prevention. So the participants in the vaccine group and the participants in the placebo group showed the same level of HIV infection. So this was really sad, and this trial was halted in 2020 as there was no sign of HIV prevention shown. We were still hopeful because we had 2 trials still ongoing, and these were the HV10705 and 706 efficacy trials. What was different about this trial was that we used an adenovirus vector at 26, and many of you might be familiar with this. This is the same one that's used in the J&J COVID vaccine. And there was also a mosaic of different subtypes included in the vaccine to make sure that most of the subtypes were covered. So we're hoping to actually see increased coverage in terms of subtypes circulating in the world, and actually raise an immune profile that would be applicable to all the global pandemic. What we saw, though, was that, in addition to this, we also saw that in non-human primates, there was good protection, and this gave us real hope that we're actually going to see a positive results in these trials. So the HV10705 Imbokodo trial was in South Africa, and Sub-Saharan Africa, and then the Mosaico in the Americas. And sadly, again, there was no sign of prevention efficacy seen in both trials, and these were stopped prematurely because there was no benefit realized. I know this sounds like bad news, but we're still hopeful in the field, and this is supposed to be... Oh, sorry. Okay. We're still hopeful that we are going to get a vaccine in the near future. So why are we interested in vaccines? I mean, yesterday we heard lots of talks about how thousands of people continue to get infected daily despite the many different modalities that we have. And we know that from time immemorial, vaccines have been shown to actually give protection to many diseases. And vaccines don't rely on adherence, so we're hoping that if we get a vaccine, we'll give it once or twice, and people will have immunity for the rest of their lives, of course with boosters. And again, if you get a good vaccine, it may end up being more cost effective than having many doses of different other modalities. So yes, we've seen a long road to an HIV vaccine, 40 years as Tim has said, but why is this? I mean, I have family members asking me, why are you still working on HIV? Why are you still trying to find a vaccine? COVID vaccines came out in like a year or so, but what's it with HIV? And there are many conspiracy theories out there. But then we know that intrinsically HIV is different. It's a different virus. It mutates very rapidly. There are many genetic subtypes all over the world. So you can't have a one-size-fits-all. HIV integrates into DNA, so it's difficult to actually remove it from the system once it's there. Melissa has just spoken about shock and kill, where you find HIV in reservoirs, and those are really difficult to actually eradicate. There's no natural recovery that has been seen for HIV, in HIV, so we don't have a correlate of protection to actually look at and do reverse engineering to come up with the vaccine. And there's currently no reliable animal model. And HIV annihilates all the cells that are meant to actually defend the body. So that's another tricky situation. But in spite of all this, I've spoken about active immunization and active vaccinations in the past, and these were meant to elicit non-neutralizing antibodies, because that's what we saw as a correlate of protection in RV144. So now we're pivoting a little bit, and we're looking at broadly neutralizing antibodies, because we know that the most successful non-HIV vaccines that we've seen around or globally are those that actually use neutralizing antibodies as a correlate of protection. And we know that in the field that broadly neutralizing antibodies, if given before a challenge, they've been shown to actually protect from HIV infection or HIV infection in non-human primate models. So we're hopeful that any vaccine that will induce BNAbs will be one that will offer high efficacy. So how do these neutralizing antibodies work? So I'm just showing here a schematic of the HIV virion. So that's the little circle there. I don't know if my pointer works, but it's the little circle there, and then the green are the little HIV ARMV spikes. So in coronavirus, we'd say those are the spikes, and here we have our ARMV proteins. And I've just highlighted and enlarged the HIV trimer in green, and you can see the antibody binding there. So if you have antibodies binding, that prevents infection of cells by that virion. And if there are no antibodies, then infection occurs. So over the years, we've been able to isolate many broadly neutralizing antibodies, and broadly neutralizing antibodies are the ones that not only neutralize the autologous virus that's in the host or in the body, but can also neutralize viruses that are in circulation in the population. So over the years, through different methods and how we've actually improved on isolation methods, we've been able to isolate different antibodies that target the different sites on the HIV ARMV, and we've been able to actually map these different sites to actually come up with wonderful immunogens. The interesting thing about BNAbs is that not everyone is able to elicit these broadly neutralizing antibodies. These BNAbs develop rarely in people living with HIV. We see them in only 20% of individuals who are living with HIV. Our study has shown this, and many other studies in the world, and it takes years of infection to actually come up with broadly neutralizing antibodies that are potent enough and have a wide range to actually neutralize many strains of HIV. So over the years, we've been able to look at this in individuals through sequencing of the antibodies and sequencing of the virus to see how the virus changes or how the antibodies change in the presence of different viruses as well. So as you can see at the bottom, you have a native or a naive antibody that we call an unmutated common ancestor, which is stimulated by that one HIV virus there. And once it gets in touch with that, it mutates and starts getting more affinity matured, and every round of that interaction with the virus and the antibody results in a broadly neutralizing antibody that can target many strains of HIV. So we've seen this in infection, and this is the very technology that we want to use to now induce BNAbs in the HIV field. So what we see here is exactly what I've shown previously, where on the left-hand side you see the breath, which is actually going up, and you see that with breath you get your development of the broadly neutralizing antibodies stepwise, and they correspond to the different virions on the right-hand side. We are going to harness, or what we're doing right now in the HIV field is harnessing what we see in infection to design immunogens that will drive a naive immune system to generate these broadly neutralizing antibodies. So what I'm showing on the right-hand side is now a priming vaccine in the green that corresponds to the immunogen that stimulated that UCA on the left-hand side. And now you can see that green spike at the bottom that you immunize with. And then the subsequent immunizations are the subsequent viruses that the antibody saw in the body, to the point where at the end you end up with a broadly neutralizing antibody that's not in just 1 out of 4 or 1 out of 5 participants, but then now you'll be able to induce these BNAbs in people who will be getting a vaccination. I know this sounds like a pipe dream, but it's actually happening. And today I'm going to show you an example of an immunogen that has been designed and has actually gone through many stages over the past 10 or so years. And this immunogen is called EOD-GT8, and it's in clinical trials, in phase 1 trials right now. But it started many years ago where a nanoparticle that's HIV-ANF-based was created to stimulate antibodies that look like VRCO1. So they have the same genetic material as VRCO1, and in there, then you can actually give the immunogen, and with the subsequent vaccination, you drive the antibodies to become more and more broad and potent in terms of neutralization. Actually, right now, we're looking at breadth. So this is what we are aiming to see in the next couple of years. So recently, a few months ago, actually, a human trial was conducted with this immunogen. And as you can see, it was given to human beings in a small trial. And when you look at the stimulation of antibodies, you can see that in flow cytometry, which is actually that panel on the right-hand side, you see that with that immunogen, you were able, or these collaborators were able to actually elicit antibodies or cells that were specific to this EOD-GT8 immunogen. And when they isolated these cells and looked at the genetic material and sequenced the antibodies, they found that in the participants in the treatment arm, or who got this immunogen, you could see those VRCO1 class antibodies that we're looking for. And in contrast, with the placebo group shown on the right-hand side, you only saw a few individuals with those specific antibodies, showing that the priming of that with that immunogen gave rise to these VRCO1-like antibodies. So this has really given us hope that we can induce broadly neutralizing antibodies in these future trials that we're working on. So it's a sobering thought, but I just wanted to highlight the fact that this didn't happen overnight. It didn't happen in 5 years. It has taken more than 10 years to get here. These are all the papers on EOD-GT8, which started many, many years ago, and that it has taken so long to actually get to a point where we say, yes, we are actually in a position to give people an immunogen that will give rise to these broadly neutralizing precursors. But the good news is, and here I'm just showing the road to this vaccine that we're seeing right there at the apex. We are 2 steps behind right now. If you can actually, I don't know if you can see that, but it's very blurry. But what I'm trying to highlight is that we are not there yet, because we haven't gotten to a point where that BNAb has been taken all the way to what we saw in individuals. So these have the mutations of an BNAb that's a precursor, but it's not the full BNAb. So to do that, we still need to guide those antibodies to get to that stage. We still need to give another immunogen to mature that antibody. But we are hopeful that now, as the technologies we know the technologies, this is going to get faster. And we are seeing right now that with all the experimental medicine trials that HVTN is conducting, we are hoping to get results quickly as they become available, so that we can see how to pivot, how to actually change strategies midway. We don't have to wait for many years to actually do this, but we can do it as we go along. So I've spoken about germline targeting, which is what I was presenting now on EOD GT8. But other strategies include the use of trimers to immunize, also fusion peptide priming, where you use a piece of HIV to actually, a peptide, to stimulate the body to produce broadly neutralizing antibodies. And another interesting part that is happening now is to see what modality of immunization we should be using. Should it be sequential? Should it be slow delivery to actually mimic what happens in the body and get to those BNAbs that we're looking for? I don't expect you to read each and every trial here, but this is just to highlight that there are many, many of these experimental medicine trials ongoing right now at the HVTN, and it's very exciting that all these trials are geared towards producing broadly neutralizing antibodies. And at the same time, we're also looking at mRNA platforms to look at so that we can change antigens and change strategies quickly. And we've seen with COVID that these are good platforms to use, and we're using them in HIV as well. So by the end of 2023, 17 experimental medicine BNAb vaccine trials will be initiated, and this will be targeting all the different sites of the trauma that I showed you. And this is just a summary to say that we have novel strategies for BNAb-inducing vaccines, but I just wanted to have a bit more time to talk about BNAbs. I'm humoring Nyaradzwa because she was expecting this, so I have BNAbs. So I did say we have a long road to an HIV vaccine, but we also have passive immunization, which will give us a faster way to deliver immunity. And we've seen in the past couple of years that actually this is possible. And this was shown in AMP studies recently, and if you have questions, Nyaradzwa can tell you more about this because she was the chair of this protocol, which was very successful in both in the Americas and Africa. And participants were given VeraCO1 as a BNAb, 2 doses over 22 months, and retention was remarkably high. So really, I was impressed by the volunteers in this study. The results from the AMP trial showed us that indeed a BNAb could prevent HIV infection, but this was only applicable if the viruses were sensitive enough. And we saw that only 25 of viruses in the African population and 37% of viruses in the Americas population were sensitive. And this showed us that we needed more potent antibodies and combinations to actually administer to participants to actually get to a level where we could have close to 100% efficacy or coverage. And so this spurred a whole lot of BNAbs to be explored, and we have currently a whole lot of BNAbs in clinical development. I'm just showing you how populated the slide is. You don't have to go through each and every one of them. But we also have enhanced BNAbs that are being tested, and these were generated by IAVI and are being tested in collaboration with HVTN. So there's really hope, and there's a lot that's happening at the moment. So where are we now? We can't ignore the fact that HIV still presents a formidable and a challenge in the field. And with these technologies that I've shown you and shared with you, which is just a glimpse of them, we are able to say we are actually moving forward. We are not stagnating. We're actually moving forward to finding an answer to this. And we're continuing to look at novel vectors, and we're looking at different BNAb approaches for studies. So I just want to end with on a positive note that an HIV vaccine is possible, and it will be a wonderful day when that is realized, and I can't wait to see that. So thank you very much. I'd like to acknowledge all the trial participants, and those are the most important, and the community and the protocol team leaders that have been responsible for all these trials. Thank you very much.